Disclaimer, disclaimer, disclaimer. Following hour of radio-friendly talk is on and around about the subject of science. If you are at all curious about the world beyond the pulp news cycle, you have come to the right place, for it is This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good radio show, Kirsten.
Hey, Justin.
What's going on?
Oh, things are good. It's been busy. I spent this week, I went down to the LA Auto Show, saw a bunch of pretty cars. I got very excited about the possibilities, so many possibilities for higher fuel efficiency vehicles, electric vehicles, hybrid vehicles. There's a lot of neat designs out there these days, and just it's not necessary that everybody's going to adopt the new automotive technologies right away. I mean, the gas combustion engine is going to be a favorite for a really long time to come, but the options are growing, and more companies are delving into the alternative space, and it was really neat to see. Pretty exciting.
Yeah, except that it's so far it's still dirtier. The alternatives, a lot of the alternatives are going to end up being dirtier for quite some time. The electric that they're putting into the vehicles, if it's a plug-in, any type of plug-in electronic is running off of coal. I mean,
no, but that's not necessary. No, but when you talk about the the well-to-wheel efficiency, so is it coming from coal? Is it coming from oil, petroleum? What's the source of the electricity? The best thing that we know about electrical is that if it's coming from a plug, that's not necessarily coal. Some areas have predominantly hydroelectric. Some areas do have wind or solar or other alternative energies that go into them. Not everything is coal. And coal
it is.
And the fact that you're taking the energy production from each individual vehicle and putting it into, the one utility, it's coming from wherever the utility is getting it from, that reduces the dirtiness of it. I mean, I'm not going to say that coal is clean, but in the sense of reducing the amount of gunk that you're throwing into the air, there's a lot more control that can be had at a coal plant than on every single tailpipe.
Well, that is a good point. The every single tailpipe thing. I still think though that coal is a much dirtier. Even a coal-fired power plant, I don't think comes anywhere near the efficiency of a vehicle today. I don't think it's anywhere close. But
yeah, I mean, I think that there's a lot of work there's tons of work that still needs to be done. Faux show.
And I've got to find this story because I heard it blurbed earlier this week that somebody was predicting a 100-year gap.
In what? What do you mean?
100-year gap between running out of oil is something we can use on a daily basis, sort of a thing, and the next alternative fuel that really comes in behind it.
Really?
So it's like almost like predicting 100-year, not completely Stone Age, but
pretty close.
Kind of Stone Agey times.
You're gonna run out of gas.
Well, it's not. I mean, gas is almost the one that we could figure out the easiest.
Yeah.
Really. The bigger impact is going to be on everything else that we use oil for, which is everything. Plastics. It's in every form of agricultural. Application. It's in absolutely everything. And tires. Each tire is what, 8-9 gallons, 10 gallons of oil?
Don't they just make tires from other dead tires yet?
No, they just make big big landfills out of the old tires.
Yeah. Well, let's get into the science news for this week. I don't need a 100-year gap here. It's here. It's now. I have stories about snot cocoons. Love this one. This week in pregnancy, surprise, surprise, and Hayabusa. We have a Hayabusa update. Yeah.
Yay.
What did you bring?
I have oil will run out 100 years before new fuels developed study. I have bacteria using toxic darts. And what is this? I have something else here that I don't have a headline for yet, but we'll be reading.
Okay, I'm looking forward to that one.
Oh, yeah, they may have isolated the molecule in alcohol that makes it so beneficial. Yeah.
Okay, be interested in hearing that one. Okay, let's talk about Hayabusa. Hayabusa had caught some dust. They confirmed it. JAXA, the Japan Aerospace Exploration Agency, just released a press release saying that they have used a scanning electron microscope to analyze samples collected with a special spatula from the sample catcher compartment A. And they identified about 1,500 grains within those samples, rocky particles, most of them judged to be of extraterrestrial origin. And
Wait, what? What? They're from outer space?
Yes, and definitely from asteroid Itokawa. Yeah, so Hayabusa, the little probe that could, went out, ran itself into an asteroid and came back with dust.
A couple years behind schedule, too. It ran into a lot of trouble out there.
Yeah, seriously.
It was a rough trip.
But it came back, and now they've been looking at these samplers and the containers. What they have in there. Most of them are little tiny 10 micron or less particles. These little grains have a number of different sources. The minerals. Let's see if I can find the. I'm missing the. Olivine is the mineral that exists the most and what they recognized. They also recognized pyroxere, plagioclase, and iron sulfides among other rock types. But the rock types and the isotopes that they found in them suggest that they were not of this earth. This is great. Now they can look more deeply into it and go, what was this asteroid made of? So we might be figuring out a little bit more sometime soon. I was just excited. A Hayabusa update. Yes. Give me some more news.
Karen Zeitvogel is the author from AFP. The world will run out of oil around 100 years before replacement energy sources are available. If oil use and development of new fuels continues at the current pace, U.S. study warns. Somewhere recently, I also heard a statistic that the alternative fuel spending by the federal government, the money they've pumped into us tracking down these new fuel sources, is somewhere in the neighborhood of $300 million a year. Which would be a lot of money for you and me to go down to Vegas for a weekend, for a month, for a year, for the rest of our lives. But for all of the alternative science research that we're doing, federal government contributing somewhere in the neighborhood of $300 million is laughable. But this is researchers at the University of California, Davis, used current share oil prices of oil companies and alternative energy companies to predict when replacement fuels will be ready to fill the gap left when the oil runs dry. Study's findings weren't very good. If the world's oil reserves were in the 1.332 trillion barrels estimated in 2008, and oil consumption stood at 85.22 million barrels a day, growing yearly at 1.3% per year, oil would be depleted completely by 2041. says the study published online last week by Environmental Science and Technology. I've heard estimates that put that 2041 much closer to today's date, but this one is saying, oddly, somehow based on the strength of the oil companies, which I think is kind of odd. But by plugging current stock market prices into a complex equation, how in the world do you figure that's related? It seems like the closer we get to it, the higher their stock prices would go, because the more money
The
they
more
could
money.
possibly
Yeah, the more
be
the
demanding
oil would
for it.
cost. Yeah.
UC Davis engineering professor Debbie Niemer and postdoctoral researcher Natalie Melishinka, Shikina, calculated that viable alternative fuel will not be available before the middle of the next century. The researchers analyzed the share prices of 25 oil companies quoted on U.S., European, and Australian stock exchanges, and 44 alternative energy companies that produce fuels such as ethanol, biodiesel, and are developing fuel cell batteries, propulsion systems, all the sort of slew of green tech companies that are out there. What they found is that the market capitalization, or total value of all stock shares of traditional oil companies, far outstripped that of alternative energy companies. That indicated to them that investors believe oil is going to do well in the near future and occupy a larger share of the energy market than the alternative energy.
Yeah, well, for the time being, oil is still cheaper. So yeah, investors are going to be placing their bets on these on oil rather than alternative fuels. Governments aren't subsidizing alternative fuels as much as they could be. Yeah, I mean, alternatives really just aren't there yet. They're
Right. Well, no, it's just there isn't.
about 100 years.
There isn't currently a foreign country out there that's sitting on a vast wealth of alternative energy that we can go and bomb or have a regime change in. So yeah, this works different, I think, than our government is used to handling energy issues. It's a different.
Yeah.
It's a complete different. It's a research problem. It's a it's a science problem.
Yep.
Not
Science, engineering, technology, implementation. That's the problem. I mean, it's going to be, right now we rely a lot on oil. So it'll be changing our reliance on oil. How can we change the products that we use? What other things can we use as in terms of oil as a fuel source? How can we create devices, cars, other devices that don't use gas as a fuel, that use other things?
Mm-hmm.
So it's demand, supply, all that kind of stuff. But 100 years, I don't know. Whatever.
The number is actually higher than I thought. 2/3 of crude oil is used to produce gasoline and diesel to run vehicles. I actually thought it was much lower than that. I didn't realize it was 2/3 of it.
Yeah. It's quite a bit. So just in case you break your spine sometime in the near future, this story was sent in by Gord McLeod. Researchers are looking at primates for a new model of spinal cord injury repair. And it turns out that axons are very, very much more able to repair themselves than previously thought. Most models of damage repair when it comes to spinal cord injury have been researched in mice and rats, and mice don't regrow their spinal cord neurons, or they do very rarely. So the common idea is that, once you get a spinal cord injury, it's going to be very, very hard to repair it. The nerves aren't going to regrow. And that even in humans, we find that some types of injuries do over time repair themselves, and the neurons do grow around the areas that have been damaged, and people regain function once again. It just takes a long time for those cells to grow. They have to move, and it's kind of hard grow those nerves. Anyway, this research paper that was published in Nature Neuroscience is pretty exciting because researchers damaged the the spinal cords of some rhesus monkeys and then looked at how they repaired themselves. They found reconstitution of fully 60% of the pre-lesion axon density that arose from the sprouting of spinal cord midline crossing axons. And so these axons sprouted out from prior to the area of the lesion and grew around the lesioned area. The animals had improvement in muscle recruitment, hand function, locomotion, and this might be, the researchers say, the most extensive natural recovery of mammalian axonal projections after injury ever observed. And so they're saying that more research needs to be done, that we need to look more, that since we're seeing this in primates, we need to look at this a little bit more closely and go, okay, what's going on there? And does this play into recovery in humans as well? Can we figure out how to heal people a little bit better? And then another story that kind of tied into this one that was sent in by Dale Fisher and also Ed Dyer. It's about stem cells from muscle tissue being transplanted into the damaged, injured muscles of mice. Basically, the researchers created Mighty Mouse out of the injured mice. When they transplanted these muscle stem cells into an injured area, they found that the mice under the cells themselves underwent a 50% increase in mass and a 170% increase in size. And that didn't go away after the repair had taken place. It actually these muscles stayed big. Mighty Mouse over the lifetime of the mice for about 2 years. So it's kind of cool that just some injections into the damaged muscle repaired and led to even bigger muscles in these mice. People who are thinking that, oh, hey, I want to get bigger muscles. I'm going to inject some muscle stem cells into my biceps so that I can be a little bigger, looking in that direction. Bad news for you all. The effect did not appear to be noticed. It didn't happen if the injections were made into healthy muscle. So there's something about injured muscle and that injury environment that seems to stimulate the stem cells to grow. So
right, and it's also, that's what you do when you're working out, is you're destroying your muscles.
Right. Yeah. That's a good point. Absolutely.
And then that's when they regrow and form other. Yeah. Very cool.
Yeah, that is a really good point, actually, that you are damaging the muscles every time, rip, every time you go through those repetitions of weightlifting. If they can figure out, this is a mouse study, so the research needs to be done, see if they can do it in either humans or see if they transplant human cells into the mice, if that has any kind of effect, if we can transplant other animal cells and see if the same effects hold. That's the next level of the research. And then eventually, if things go as they hope they do, see if it works in people and maybe be able to find out the signals. It's the kind of
athletes that we've been waiting for.
No.
So we can have
to help people with muscular dystrophy.
70, 80 home runs a year, like on the average. Like the low end guy is going to be hitting like 80 home runs a year.
Right.
That's because that's the exciting part of like the sport,
Yeah.
We want to watch all these games where people are hitting singles and trying to run base. Nah, you want people knocking the ball out every time they're up there.
Out of the park, Mighty Mouse. That's right. That's right. I'm thinking maybe let's try and help fix muscles of people with muscle disorders. That might be.
Yeah, but they're not going to hit home runs, Kirsten.
Maybe they're
not going to generate the kind of excitement that McGuire and Sosa did.
I don't know. Maybe they will. If their muscles are big, no roids involved, could be good. Could be good. What you got? This is This Week in Science, everybody.
What do we have? We have bacteria. In nature, it's a dog-eat-dog world, even in the realm of bacteria. Competing bacteria are now using toxic darts to disable each other. Like, now we're using as though this is a current event. This is just started
now.
happening.
Just
started
happened.
doing this.
No.
According to a new study by UC Santa Barbara biologists published in a Nature journal, the discovery of toxic darts could eventually lead to new ways to control disease-causing pathogens, says Stephanie K. Akoy, first author of postdoctoral fellow in the UCSB's Department of Molecular, Cellular, and Developmental Biology. This is important because resistance to antibiotics is on the rise. Oh. So they're going to look, I guess, to find a way to hire some of these toxic dart-throwing bacteria to do our bidding.
I can just imagine the job selection process. Excuse me, Mr. Bacteria, how is your aim?
First, we need to learn the rules of this bacterial combat. It turns out that there are many ways to kill your neighbor in the bacterial world. They have a wide range of exotic toxic darts to choose from. Scientists studied many bacterial species, including some important pathogens, found the bacterial cells have stick-like proteins on their surface with dart-like tips. These darts are delivered to competing neighbor cells when the bacteria touch. This process of touching and injecting a toxic dart is called contact-dependent growth inhibition.
That's
CDI.
quite a title for. I killed you with my toxic dart.
I didn't kill him, Jan. It wasn't murder per se. It was contact-dependent growth inhibition. Is all that was going on there.
That's right.
Some targets have a biological shield, though. See, there's already we're talking predator prey, countermeasure, bacterial level. God, I love bacteria. They're so awesome.
I know.
Bacteria protected by an immunity protein can resist the enemy's disabling toxic dart. The immunity protein is called the contact-dependent growth inhibition immunity.
Yeah.
The protein basically inactivates the toxic dart. The team discovered a wide variety of potential toxic tip proteins carried by bacteria cells. Nearly 50 distinct types that have been identified so far. Each bacterial cell must also have immunity to its own toxic dart. Otherwise, carrying the ammunition alone would kill them.
You don't want that to backfire on yourself. It's like having a safety on the gun, or you have to find a way to make sure to keep yourself alive while getting rid of the competition.
Right. Yeah. It's like it's like the pox in the early American when we were giving pox-covered blankets to the Indians.
Right.
We already had the immunity, and it seems like a very modern world devious mass destruction, but apparently this has been going on since the days of bacteria.
Exactly, the bacteria. Jeremy in the chat room was wondering about was asking if darts are more effective than just filling themselves up with toxins until they explode. And I guess, to self-preservation, it's a lot better to just have a dart that gets rid of your neighbors than having to actually go terrorist on everything.
Yeah, that's the equalize method,
right?
Which is to develop the toxin, share just a tiny bit of it with enough of its own colony so that they all have an immunity to it, then go over to the neighbors and explode themselves so the toxin goes everywhere, kills off all the neighbors, their own colony can move into the area and is immune to the toxin, so it doesn't matter. Suicide bombing bacteria.
That's right. Wow. That's pretty cool.
So really, when we have like a pathogen, a bacteria that's causing all sorts of infection or something in us as humans, it may actually be waging war on us. It may actually be trying to kill us.
Technically. Yeah. Technically, yes. I think most of the time, though, we are, beyond the plane of their their lifetime comprehension, whatever comprehension bacteria have. We're just collateral damage. for the wars that bacteria wage with each other.
Yeah. I wonder if the
know?
Earth thinks of it that way. The Earth is like, yeah, you can't really blame the humans for ruining things. I mean, they're just not on the same plane of existence as a planet.
No, they're not. Exactly.
Short-sightedness.
Yeah, well, speaking of maybe more long-sightedness, there's this great news out of CERN this last week. Researchers at CERN, where the LHC is housed, have trapped antimatter. 38 antihydrogen atoms have been caught in a magnetic trap. Yeah.
What were they trying to do?
Well, the physicists at CERN were actually trying to catch antihydrogen. Antimatter annihilates with matter when they come in contact. So in our universe, the majority of matter is matter. Antimatter, just we don't really see much of it at all, and it's hard to keep around because it annihilates when it comes in contact with anything. So they had to try and capture. They had to be sneaky and try and capture these antihydrogen particles. According to Ars Technica, these particles behave predictably in the presence of electric or magnetic fields, and so they can use a special container called a penning trap, which is exactly what they used to be able to contain the antihydrogen. However, antihydrogen has no net electric charge, and so they had to go to a little bit more complicated measures to get them. So they super cooled the antiproton decelerator. They basically used let's see what they did here. They used a superconducting magnetic trap that would interact with the antihydrogen's magnetic moment and thus trap the antihydrogen inside of its inside of the area, confine the antihydrogen at the ground state. And so the temperature that they had to super cool it to was less than half a Kelvin, which is pretty cold. It's really cold. The antiparticles, these antihydrogen, the use of them, I guess, is to start figuring out the underpinnings of the standard model of physics, really, to be able to figure out how physics is put together. Are we on the right track with our ideas about the way everything works, or are we not? And can we use these particles to find out exactly why there are more regular matter particles than antimatter particles? Because we are definitely not a symmetric universe in this particular way. Even though there are so many things that do seem symmetric about our universe, this is very very interesting to scientists, and they want to find out. So they want to test these symmetries in nature and find out what happens if they excite the antihydrogen into various states of matter. One of the interesting things about this, antimatter is science fiction. We love the idea of antimatter for warp drives. You could get, when matter collides with antimatter, there is a huge amount of energy released, and that huge amount of energy could be enough to power a spaceship through outer space if you could create a warp drive. However, the number of collisions that we've had and that we've seen, the reason that we're not collecting this energy all the time is that there really aren't that many antimatter particles around. It's hard to keep them around, and it's hard to put them in the exact place you want them to be in to be able to collide and use that energy. So currently, we basically have to have giant accelerators to be able to smash protons at each other to be able to get these particles out. And then we have to have these special magnetic traps and the supercooling environment to be able to actually trap these particles. So right now, this is just proof of concept, and they've got them, and they've done a few little tricks where they've let the antiparticles kind of out of the trap to let them collide with stuff so they can actually prove that they're there. The only way that they can prove that they trapped the anti-hydrogen particles is to destroy them. So it's just proof of concept, so that next time they can be like, we know we got them in there. We don't have to destroy them now, but we know we have them in there. So anyway, warp drives in the future. It's a possibility.
I think it's just an excuse to see stuff blow up.
At a very, very small scale.
Really? We've got antimatter particles in here? Really? Yeah.
Really?
Can we blow some up? How many? We got lots, right? We got plenty.
We've got 38.
36. No, it was 36, right? 28. No, it was 28. That's how many we started with.
Yeah. So somebody in the chat room is wondering, like, this is not the first time that antimatter has been created at a collider lab. Lots of colliders create antimatter all the time. This isn't even the first time that they think they've trapped it. This is just the first time that they've really proven that they trapped it, and that they got these anti-hydrogen particles. So, antimatter, and this is just one type of antimatter particle, the anti-hydrogen particle. And so this is just one particular instance. Yeah. Interesting stuff. It's time for us to take a break. CERN traps.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science.
You like audiobooks? If so, you can get yourself a free one. We always like free things, right? Audible.com is a leading provider of audiobooks with over 75,000 different titles in a variety of genres. Twis has found many science-based books in the Audible library, and you can start a free trial today and get any audiobook download of your choice for free. All you have to do is sign up at audiblepodcast.com/Twis, TWIS. It's just that easy. Go to audiblepodcast.com/Twis now for your free download. And you can read Bad Science by Ben Goldacre with the Twis Book Club. That's right. Bad Science by Ben Goldacre is the November Twis Book Club Book of the Month. And we're enjoying it quite a lot as we move forward and discover, all the wondrous ways that science is getting misused these days. Head on over to twis.org and get yourself a copy and join in the conversation before the end of the month. We still got some time. We still have some time. Join on in.
It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. This Week in Science. Science. I've got one disclaimer, and it shouldn't be news, that what I say may not represent your...
And we are back. This is This Week in Science, the hour of science that, we try and fill your week, let you know what's been going on in the science world for the past week. We try and get in there and help you out this way. Story sent in by Tony Steele this week that I really enjoyed. Metal. Did you know that metal doesn't really have any color? That the electrons within metals usually absorb and emit light over the entire optical range. This is from an article from the Physics Archive blog. And so silver is the way it usually kind of comes out to our eyes, but metals themselves are actually colorless. It's kind of interesting to me. So researchers are figuring out how can we make them have different colors. Gold is a color that we're familiar with. The reason that gold is because it absorbs blue light and it gives off a yellow color. Copper absorbs blue and green light, and so then you see the reddish orange color of copper. So some metals actually because of the way they absorb and reflect the light, that's what you see in terms of their color. Now, how can we actually give them a different color? Some researchers at the University of Southampton have decided that they can do it by carving nanoscale patterns on the surface of metals. Their idea really is you could make jewelry that it's kind of techno jewelry and would have it's gold, but it's purple gold instead of the yellowy gold that you would see as a result of the etching that they would do on the surface. And so they basically etch these nanoscale circles on the surface of, say, gold or whatever metal they choose. And the spacing of the circles and the depth at which the circles are etched will change the resonance of the metal of the surface so that the light is then absorbed and reflected differently. And so your eyes would see a different color. So it's a new technique that's being suggested that we could basically, as Tony put it, carve a rainbow into metals. And if you have any, there could be other ideas for what we could use it for. But so far, the researchers think jewelry.
Yeah, totally.
I think it could be fun. It could be fun. Or makeup. Maybe, yeah.
Yeah, they're using, or even, I don't know if tattoos would really come into play there, but they're using some sort of fine metals that they've crafted in the way that that really blue butterfly has that luminescent blueness to it.
Yeah.
And they're using that now in makeups. And I think they were using some sort of metal particles to pull that off.
Yeah, I think so. What you got? More stories? Got more news?
We've covered I think I've mostly covered many studies purporting the benefits of drinking and how it's beneficial when it comes to cardiovascular health. First time now, scientists have discovered that a well-known molecule called Notch may be behind alcohol's protective effects. Down the road, this finding could help scientists create new treatment for heart disease that mimics the beneficial influence of modest alcohol consumption without the modest alcohol consumption, which sort of sounds like they're looking for a cure when they already have one. Like, I don't know. Any understanding of a socially acceptable modifiable activity that many people engage in, like drinking, is useful as we can continue to search for new ways to improve health. See, it's already socially acceptable. It's already a modifiable activity that many people engage in.
Yeah, it's socially acceptable until you drink too much. As long as you're in the moderate range and you're still maintaining socially. It's the socially acceptable, lubricant that it feeds conversations at bars and awkward conferences and stuff.
Yeah. Makes people feel good.
Feel great. So I'm going to have another. I'm going to have another. Oh, shouldn't have had
Next thing you
another.
know, I'm saying stuff I'm probably going to regret later. Yeah.
So,
We're doing things. So if we can figure out the basic science, on the basic science level, how alcohol is beneficial, it wouldn't translate to doctors prescribing people to drink, but hopefully will lead to the development of new therapy for millions of people with coronary heart disease.
right.
Yeah, why aren't doctors recommending that people drink?
Well, drinking itself is not necessarily, there are other problems that come along with it. There are benefits, but they're thought to be benefits to cardiovascular health if it's done in a moderate manner, but often
Right.
it's difficult for people to maintain in a moderate way. And so,
Sometimes.
if you could just not drink at all.
But isn't that true of
and-
any drug that's prescribed? I mean,
Yeah.
but I mean, I've never heard that there was a doctor saying like, well, have 2 drinks tonight, call me in the morning, see how you're doing.
Right.
If you're feeling a little better, have a couple more drinks the next night, work your way up until you're doing like 5-6 drinks a night. And then, you probably should stop right there. But I don't think they are. I don't think they are. Even the people who have cardiovascular health issues are telling them to go out and exercise. No, there's other ways. I don't know. It just seems like it could be something that could be prescribed. I've just never, and maybe it is. Maybe that actually happens. I've never heard of it though, which is kind of interesting.
That you haven't heard of it?
No, it's
You
kind of
hear of
interesting.
everything.
Well, it's kind of interesting that we have a medical system that prescribes so many different narcotics.
Right.
That are from the industry of a narcotic that's only being created for that. But you don't have, marijuana being prescribed now for glaucoma and for some other, sort of things.
And there are other,
marijuana is starting to take place.
and there are other drugs that are based on the cannabinoid receptor, the THC molecule. There are drugs that have been developed and are being prescribed for various things, and it's not, not go smoke a joint and call me in the morning.
No, but still, I wonder why. Like, I've at least never heard of a doctor prescribing alcohol.
Yeah. Well, I think that there's nothing specific that alcohol, having a drink, will not specifically affect, certain disorders like marijuana, glaucoma, or nausea. We know that there are direct results to the imbibing of that drug to those disorders. But with alcohol, there's no real. It's just if your lifestyle involves a glass of wine a night, you're probably going to have better cardiovascular health overall. There's a link. It's a correlation. It's not a direct result.
Then
you can't prescribe
if it's not a direct result, then why are they isolating the molecule that might be responsible so they can refine it into a drug that a pharmaceutical company can produce?
It could be linked directly.
because it's not a pharmaceutical product.
Right.
It has no merit. It almost is like a holistic drug then, alcohol. It really is. It's
dear.
No. I'm not drinking. I'm just engaging in homeopathy. That's all this is. It's just holistic medication. That's all I'm doing.
Right.
Yeah, the doctors won't tell you. It's one of the big secrets doctors try to keep from you.
Oh my goodness. Well, one of the things that fish, particular reef fish, parrotfish, wrasses, some others among those reef fish, they tend to wrap themselves up in a cloak, a cocoon, so to speak, of snot, of mucus when they go to sleep at night. When they bed down for the night, their blanket is not a nice fuzzy thing. It's made of snot. And researchers have been trying to figure out exactly why Why would they wrap themselves in a blanket of snot? So they did a study, and they put parrotfish in some tubs, and they let them go to sleep at night, and they scraped the mucus off of some of the fish. And then they put this terrible parasite that sucks the blood of the parrotfish, kind of like a mosquito, actually, into the bins with the parrotfish. And the fish that still had their mucus were less bitten than the fish that did not. And it was significant. So 94% of the fish that had their cocoons removed had bites. 10% of the fish that still had cocoons had no bite or had bites. So the number of bites were definitely related to wrapping up with that nice cozy snot blanket before bedding down at night. It's just I think just a really, really interesting study about what animals are doing with what different animals do to protect themselves and why they do it. The mucus is not hard for them to produce. It's very energetically cheap, so it's a lot easier than them having to do energetically demanding behaviors like scratching themselves on rocks or staying away from particular areas that might have food but might have lots of parasites in them. Another point that's brought up is having to actually actively seek out the fish that feed off of parasites. So you've got a parasite sucking your blood. You're like, I gotta go find the cleaner fish, get this parasite removed. It's pretty interesting. Mucus is used for so many things in the animal world. It is amazing.
I have now a new plan if I'm, happen to have a cold and there's a mosquito in the room at night.
I know. Protect me from the mosquito, my blanket of snot.
But is that really any stranger than early human beings like skinning other animals and using their skin wrapping their skin around them to, ward off bites?
No, not necessarily. I mean, it's pretty energetically demanding to go hunting for an animal and skin it and then use their skin to protect you. Other things that humans do are go searching for herbs and other plants that they can rub on themselves so that they're aromatically protected. I don't know if that would necessarily work in the water. I don't know if that would work. But yeah, different animals have different methods of trying to protect themselves from those pesky bites. Give us another story.
Okay, schizophrenia, we know, is a placeholder mental disorder. It's a bunch of different sort of disorders that are sort of wrapped up and haven't been isolated and removed from the big schizophrenia pool of disorders. But as it is, still, it is being called schizophrenia. And they're finding that it differs between the sexes in terms of age at onset, symptomology, response to medication, and the actual structures of the brain anomalies. Now, a new study from University de Montreal shows there's a gender difference between men and women's mental ability, with women performing better than men. These findings published recently in Schizophrenia Research have implications for more than 300,000 affected Canadians, this being a Canadian study. We are the first to report sex differences in brain function of schizophrenics, says Adrian Mendek, researcher at Central it's a very long French word, hospital. It's La Fontaine. We chose to study a task involving mental rotation of a three-dimensional image because in healthy men and women, this consistently elicits differences in terms of reaction time and performance accuracy. The results were opposite for control versus schizophrenics. The Mendrick and her colleagues compared the brain function of healthy volunteers with schizophrenic patients that were completing the image rotation task by using magnetic resonance imaging. Their findings confirmed that healthy men performed better than healthy women. This is sort of one of those generalizations that may, have some
factoids to it through how we mentally evolved over time. But men have a tendency to be better at spatial relationships.
Right. That's a bias.
It's a bias. I don't know if that's actually true. According to their healthy volunteers, their control group, that's the way it came out. Working with this spatial object, the men were better at it than the women. But when they tested the schizophrenics, they found the women performed better than the schizophrenic men.
Oh, that's interesting. So the effect was reversed.
Yeah.
And did they were just looking at how they performed, and so they don't actually know why? Did they look at the brain regions involved at all?
I haven't read that far yet.
You're a big help.
Sex hormones such as testosterone and estrogen may explain the results, says Mendrick. Findings from our other studies have shown that the testosterone level was positively correlated with activity in healthy men, but not in healthy women. And conversely, schizophrenic women have been shown to have higher levels of the hormone. Interesting.
I wonder what that. I just wonder what that means. So hormone differences are kind of reversed, but what does that mean for the development of different brain regions that would be responsible for those particular behaviors? Is there, I guess the question would be whether or not those hormonal differences are present from youth or puberty. I guess puberty when it's after with puberty. That's often when the symptoms of schizophrenia start kicking in, right?
Correct. And it can be much, much earlier than that, though.
Yeah.
Then the question is it presence in the womb, in the development?
Right.
Is there
it something
a
that's
different
early?
level that's going on there that's affecting what those levels will be much, much later as well? But because, it's largely a lot of schizophrenia does really become apparent in the teenage years when it becomes harder to maintain, a sense of normalcy. But it actually does manifest much earlier than that. It's just not as obvious and it's harder to detect earlier.
Right. As like so many things are. I mean, development is so,
And children, it's so hard to tell.
Complex.
But, I mean, children are just almost purely delusional creatures. They are.
That's true.
I mean, you can't get away with. I couldn't just all of a sudden start a conversation with my imaginary friends here and go on with it for half an hour.
Right.
Without,
people thinking you had something going on. Something a little strange.
They get away with it. They can They can just be in any world, planet, place at any point in time. Doesn't matter. They're fine with it. So, not being able to escape that world, having those invisible friends, those imaginary friends, have their own sort of vitality in your brain where they are, in a sense, it's almost a separate part of your brain is driving their conversations. They're the imaginary end, where children are pretty much completely in control of what their imaginary friends are saying and doing, what their names are.
Yeah.
What the entire field of play is up to them. What happens if you have delusions and schizophrenia is that the person who's experiencing this and involved with this isn't in control of all those different aspects of their brain.
Not conscious control.
Not conscious. Well, not subconscious control either. I wouldn't think. I mean, it is stuff that's going on in their mind that's out of their control.
Right. Possible. I don't know. Well, thinking about development, let's talk about pregnancy. This week in pregnancy, there were several studies that I thought were rather interesting. One study sent in by Ed Dyer talking about microRNAs being found to be the link that pretty much triggers contractions during labor. This study out of the University of Texas Southwestern Medical Center in Dallas, led by biochemist Carol Mendelson, looked at how different genes in the mouse uterus changed with the onset of contractions. They found that as progesterone levels in the blood dropped, there was an increase in specific microRNAs within a family called the MIR200 family. And within these microRNAs, there were, or basically controlling the expression of these microRNAs, they found two genes, ZEB1 and ZEB2. ZEB1 and 2 are really sensitive to progesterone. And so when progesterone's high, the levels of these genes are high, and the genes suppress the microRNAs from being expressed. And then as the progesterone drops, ZEB1 and ZEB2 get dropped as well. And so they can't control the microRNAs anymore. The microRNAs increase, and they end up actually feeding back again to further negatively to inhibit ZEB1 and ZEB2 even more. So that if progesterone came back, it would be really hard for ZEB1 and ZEB2 to get control of the microRNAs. And again, these microRNAs thus are really important in the onset of the contractions. So the researchers they looked for similarities also in other animals. They checked in cells from humans, and they found the exact same response. And then what they said were like, oh, okay, microRNAs are they get affected by inflammation. I wonder if, and inflammation can often trigger early labor. So maybe these things are all linked. And so they then took some mice again, and they treated them with one group with labor-inducing hormones that would drop the level of progesterone to see what would happen. And of course, ZEB1, ZEB2 dropped, the microRNAs went up, contractions, labor. The other group, they gave them an inflammation-inducing compound. The inflammation caused ZEB1 and ZEB2 to decrease and increased the microRNAs as well. And so the researchers think this could be the link that they've been looking for to be able to control preterm labor, so that if an infection, something that causes inflammation, triggers early contractions, that those might be counteracted by something else, now knowing what the pieces of the puzzle are. So this could lead to helping women not have premature birth as often, which could be pretty cool. Another study, this was reported in the BBC, it was sent in by David Eckerd, suggests that the placenta and the complexity of the cellular structure of the placenta and how it connects the mother and the baby could be linked to how long the gestational period is. It's not the only thing involved, but researchers analyzed 109 animal species, and overall they found the more complex and folded the placenta of a mammal, the shorter the gestational time was. So mice have this really, really short gestational period, and they have a really complex placenta. Humans have this really long gestational period, and our placenta is actually not that complex when it comes down to it. It's a nice little barrier, transfers nutrients and stuff, but it's not as complex as a lot of other organisms. Final little tidbit in here that I thought was really interesting has to do not with, I guess, not with the hormones involved or the structures involved, but has to do with how chemicals could be affecting development of organisms through gestation into behavior and onward. Researchers looking at wastewater chemicals found that fish, when they took triclosan and triclocarbon, they're antibacterial components that we often have in soaps and mixed in with plastics for cutting boards and kids' toys, that these compounds in the wastewater actually affected father fathead minnows, which usually establish nests and defend defend the nests from rivals in males that were exposed to the triclosan and triclocarbon. They weren't that interested in defending their nest anymore. Their behavior totally changed. So these compounds can have significant effects in the wild. Additionally, researchers looked at ibuprofen in water and how it affected enzymes. They found that in zebrafish, 50 micrograms per liter of ibuprofen, that's more than what's normally found in the wild. Environmental samples have been found at levels up to 20 micrograms per liter, which is about 20 parts per billion. So they were using a much larger amount of ibuprofen in the water, but they found that that ibuprofen affects, it works by inhibiting enzymes called COX-1 and COX-2. In humans, this is part of the inflammation pathway. In fish, they're pheromones. They're actually, these prostaglandins work as pheromones. So the fish that were exposed to the ibuprofen water they didn't have as much courtship behavior. The males, instead of going into courtship, they'd avoid each other.
Wow.
Yeah.
That's pretty wild. That's some resourcing of our evolutionary gene pool. That's quite a ways from what it may have been previously.
Right. Yeah, I think we were talking earlier about the effects of the bacteria on us and whether they're waging war on us or whether it's we're just the collateral damage. Well, I think this is the kind of thing that it's our collateral damage. It's, we're not waging war on the fish. We're not, out to get the environment. We like living in the environment. And these are just kind of the side effects of things that we do. And we don't even know all the ways that the chemicals.
I don't think we like living in the environment, Kirsten. Are you kidding me? Look how we live.
I live in a bubble.
I live in a cave. Okay, we all live in caves.
It's true.
They're not in mountainsides anymore. We can put these little caves wherever we want. We don't want the weather that's outside, right? So we've insulated the whole thing, and we've got the heater, cooling devices. We can change the weather. We make the weather whatever we want it to be, the temperature whatever we want it to be in here, and then. Well, we don't want to really travel on the earth from one spot to the other because it's dirty and, comes apart in the wrong places. So we pave over everything. We make our own artificial rock that we place down on everything. There's in fact, and we don't like the darkness. We don't like it to be nighttime. So we put lights everywhere. So it's never dark anymore. There's nowhere you can go in any city and it's just a dark place to be.
Yeah.
Wait, I don't think we like the environment.
Let's change it so that it's more the way we like it. Okay.
We must like pollution. We must like pollution. It must.
I don't know
must
about
be,
that one.
I think we do.
Yeah.
I think we do. Yeah.
Dirty people.
You have to say if somebody's engaged in a behavior like this, like whatever anybody's job is, whatever anybody's occupation is, even if it's the dirtiest, muckiest thing, at some point they've chosen it. At some level, they've chosen to do this. There had to be a way to do something different. And the fact that en masse, the entire planet, the entire United States is we're big polluters. We love to pollute. I don't think we like the environment. I don't think that's going to change anytime soon, except for the people who really say that they like the environment, who really claim to love nature.
You're going to have to ask them to prove it to you.
No, the people who really claim to love nature go out and they build their caves in nature.
Right.
They go and clear out some nature in the heart of where nature was, or they drive their cars through nature, or they take walks through nature where there's been paths carved that disrupt the natural habitats. So those who really claim to be loving nature are even more aggressive in going out and trying to kill it.
On that note, we're going to close the show. We're at the end of the show. Unfortunately, another hour has passed us by too quickly. I'd love to give shout outs to everyone who sent in stories this week. Monkey, Gord McLeod, Ed Dyer, David Eckerd, Tony Steele, Pamela Taylor,
Frank Lloyd Wright,
Frank Lloyd Wright. What?
He didn't
Dale
send
Fisher.
anything
There were
Thought he
some
needed
great,
a shout out.
yeah, some great stories that were sent in. Thank you very much for keeping track of the news and letting us know what you think is interesting. I do appreciate it.
Oh, and if you've you can send into our. We have emails, right? It's
Again, really?
wait.
Yeah.
Here,
You
can
know
edit
you
this.
know this. You know this, Justin. You can do this.
No, I don't have it memorized. I swear to. Thank you, everyone, for listening. We hope you enjoyed the show. Twist is also available via podcast. Just Google us in the iTunes directory, This Week in Science, or if you have an Android device, you can look us up in the Android marketplace, Twist for Droid.
That's right. For more information on anything that you've heard here today, show notes are going to be available on our website, twist.org. We also want to hear from you, so send us an email at kirsten@thisweekinscience.com or twistminion@gmail, which is Justin's new email address.
And you can also get us on Twitter. We are at Dr. Kiki or at JacksonFly on the Twittering net. We love your feedback. If there's a subject you would like us to cover or address, a guest suggestion for an interview, please let us know.
And we're going to be back here next week early. So we hope you'll join us again for more great science news.
And if you have learned anything from today's show, remember,
It's all in your head.
I've got the kind of mind I can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Woo! Another show down. Another show bites the dust, and Justin runs away. And Justin runs away. And Justin runs away. Another show bites the dust.